we are moving to microscopic plant remains, of which pollen is perhaps the most well-known one. Pollen and spores are such complex microfossils that a whole branch of paleobotany, known as Palinology, is focused only on studying them. Pollen grains are the male gametophytes of seed-bearing plants, and the spores are similar to pollen, but they exist in species of ferns and mosses. The most common way to study them is through the identification of taxa with the help of a microscope. This is possible due to the fact that every species has specific pollen grains, and also they are extremely varied in shape, size, presence of openings on the surface and their shape and position, and also then the presence of little wings, bumps, reticulated surface, and so on. This can be seen by many examples on the slide. A very vast number of plant species produces pollen and in very large amounts, so it is often recovered in all the stratigraphic layers. The grains are very small and have a strong inorganic outer surface, which enables them to be fossilized by almost all the modalities, apart from carbonization, because they don't endure strong burning. And most commonly, they are recovered from anaerobic waterlogged environments, usually by coring, which is a technique where a long hollow bar is inserted into the ground to make an uninterrupted stratigraphic sequence from which the samples can then be taken. This is such a core here on the bottom, and a simple soil samples packed into clean bags can also serve for pollen studies. The main drawback of these analyses is that it can very easily be contaminated by mother pollen, since it is present everywhere. So special care needs to be taken to keep the tools and the rooms for analysis sterile. By the use of pollen and spore analysis, reliable reconstructions of paleoclimate and paleovegetation are obtained, but we can also study land use and also detect wild plants at archaeological sites.